Thank you to the organizers for inviting me to present my work, and thank you everyone else for being here today. My name is Jordi Abante, and I have been a member of the Gutierrez Lab at Johns Hopkins University for the past 5 years. I recently defended my dissertation, and I will be soon studying my postdoc at the Salzman Lab in Stanford University. During this presentation, I will go over our recent work on a computational method for estimating epigenetic energy landscapes from nanopore sequencing data. Since I imagined there would be a myriad of backgrounds among us, I thought it could be useful to give a few basic notions about epigenetics and DNA methylation so as to provide some context. The central dogma of molecular biology explains the flow of genetic information from storage in the DNA to function as proteins or RNA molecules. DNA can get replicated during cell division, but it can also get transcribed to produce RNA molecules, which in turn are translated to produce proteins with a specific function encoded in the original DNA. It turns out, though, that genetics is not the end-all be-all, since behavior and environment have an impact on these processes. For instance, what we eat, whether we smoke or not, or even the amount of physical activity we do can affect the central dogma, and epigenetics is the branch of science that studies the underlying mechanisms. More precisely, epigenetics studies the set of chemical, heritable, and often reversible marks that regulate the central dogma. This figure illustrates the different epigenetic mechanisms that cells leverage to function. There are many different types of epigenetic marks, and today I will focus on DNA methylation, in particular 5-MC DNA methylation in CG context. Studying these marks allows us to fill in the gaps in the central dogma and to answer many relevant questions. For example, we can try to understand the mechanisms our cells use to decide which genes are used or when they are used, i.e. gene regulation. As a result, it should not come as a surprise that epigenetic marks play a critical role in a number of health endpoints, for example, in cancer. How do we go about studying these marks, though? Let me briefly discuss that in the context of DNA methylation. Traditionally, bisulfite sequencing has been the go-to technology for comprehensive methylation studies. Briefly put, the bisulfite treatment and subsequent PCR transform unmethylated cytosines into thymines. After aligning the reads, we can determine the methylation state based on these modifications. This technology, however, only allows to study 5-MC DNA methylation and produces very short reads, around 150 base pairs. Nanopore sequencing is a relatively new technology that offers a new exciting paradigm in genomics and overcomes the main culprits of bisulfite sequencing. This technology can produce very long reads. Recently, an entire chromosome was sequenced, and simultaneously contains information about several epigenetic marks. This gives nanopore sequencing an incredible edge over bisulfite sequencing in certain applications such as the study of repetitive elements or in allele-specific analysis. In this case, the DNA is unwound and single-stranded DNA runs through a pore where current measurements are taken. The basic premise being that the current measured will be a function of the chimer running through the pore. This technology, however, has an important limitation, and that is that the signal is rather noisy, and that needs to be taken into account when performing the downstream analysis. To that end, we build upon two previous works. On the one hand, we use the DNA methylation landscape proposed in 2017 by Jenkinson et al., consisting of a biology-inspired landscape energy function, which assigns a potential energy to each possible methylation pattern of the region studied. High energy values indicate improbable methylation states, and vice versa. The main idea can be seen in the left figure. In this example, the left landscape is shallow, resulting in consistent methylation patterns, here represented with balls, whereas the landscape on the right is much flatter, assigning similar energies to all methylation patterns. This model constitutes the hidden unobserved state in the proposed approach. On the right-hand side figure, we're illustrating the noise model. On the x-axis, we're showing the current values measured by nanopore sequencing produced for two different versions of the same chimer, the methylated and the unmethylated versions respectively, each one having its own current distribution. The work of Simpson et al. Consisted in estimating from real data the distribution of the current signal given the methylation state of the chimer, and that's what we will use as our emission probabilities. So we can combine these two ideas to create a hidden Markov model where the hidden layer consists of the DNA methylation landscape, here represented with X, and the observable layer consists of the current signal Y. Now, to estimate the parameters of the latent methylation landscape model, which is what we're ultimately interested in, and we're going to denote by theta, we propose to use the expectation maximization algorithm. As many of you know, the EM algorithm consists of alternating an expectation step and a maximization step, like shown here, until convergence. This process involves many computationally heavy operations, such as computing the partition function of the methylation landscape several times, and this can be a hard hurdle to overcome if done naively. To overcome this issue, we leverage the transfer matrix method, which allows us to compute the required quantities rather efficiently. There are a lot of details that I'm skipping, so please ask me later if you're interested. In any case, to study the validity of our approach, we perform realistic simulations using Deep Simulator, a recently proposed method to simulate nanopore sequencing data. So using simulations, we studied the performance of the approach in estimating the expected values of methylation frequencies, a commonly used quantity, and we compared our approach to two models that binarize the nanopore data by applying a simple threshold on the log odds of the conditional probabilities. On the one hand, we looked at the naive empirical estimator, which is what most researchers are currently using, here shown in orange. On the other hand, we looked at a model that consists only of the hidden layer shown before, so it doesn't really model the noise, and that's shown in blue. Overall, we found that modeling the noise leads to substantial gains for the various noise levels we considered, including the real noise levels, which are around 3. Here I'm showing the estimation error, so the lower the better. We can see that our approach, here shown in green, performs consistently better and it's more robust to higher levels of noise. So again, we obtain clear gains from modeling the noise in the current signal. We also studied the behavior in estimating higher order moments, such as correlations between contiguous CPG sites, and we found that our proposed method is superior across the board. This can be useful in estimating quantities like methylation entropy, for example. So here we presented the first method for landscape analysis for nanopore sequencing data. We show that modeling the noise in the current signal leads to substantial gains, and we propose an algorithm that is robust to various degrees of noise, making it very general. This would allow, for example, for a higher sequencing speed. Our approach extracts more information from the data, and it provides significant savings in terms of sequencing cost and time per read used in the analysis. This has huge implications considering that sequencing takes days and thousands of dollars. This work also lays out the foundation for the study of other epigenetic marks or even epitranscriptomic marks, and to study the interplay between chromatin organization and the epigenetic landscape. We have implemented all of this in a Julia package together with a versatile differential analysis method, which will be made public as soon as the full paper, currently in review, comes out. In conclusion, we believe that the proposed approach can enable and accelerate both basic and clinical research. With that, I would like to thank the other authors of the paper, John Gutzias, Sandeep Kambapati, and Andy Feinberg, as well as Winston Tim and Jared Simpson for the helpful discussions. And now I will be happy to answer any questions since I had to skip a lot of details in the interest of time. Thank you very much.